to categorize study designs basically. Under original studies too, can I begin to go down a bit? Are you doing what we call a cross-sectional study? Cross-sectional study is what all of us we are used to most of the time, sharing questionnaires and all of that. Then you can do a retrospective study. So if you want to do a retrospective study now, let's say you can do a retrospective study, a 10-year retrospective study on, let's say, experiences of May surgical patient in Ladoke University of. So you are looking backwards. So you have to get access to all the patient question, what do we call it? Case record for the last 10 years. So you are looking back. Then we can do cohort studies too. Cohort study is a prospective study. So we can say from today we want to be studying the patient that comes into our general patient clinic over the next 10 years. So that's a prospective study. So a lot. There are case reports too. Case report. In case report, you are trying to report novel or unusual presentation of diseases. So, and when you have a large, maybe like 5-6 case reports together, it becomes a case series. So so there are different ways. The reason why this is important is, number one, before you say I want to do research or I want to do a study, you must be able to figure out which of these study designs actually fits or is visible for me to answer this research question. So there are research questions that can only be answered by a randomized control trial. If you are testing a new drug now, vaccines and all of that, they usually use what's it called? A randomized control or clinical trial. Generally, it may not be randomized. It could even be non-randomized, but those are clinical trial based answers. So and also you want to count your cost as a medical student. Which one is within your reach? Do you have the resources of getting across questionnaires, having access to what's it called now, patient case record and things like that. So when you have a research question in your mind, the first thing is to sit down and feel like, what do I have enough resources and enough capacity, and what is the best study design this question? So that's what the knowledge of a solid understanding of study design does for you. It helps you to be able to use the right study design to answer your research questions. For instance, there are study also or other types of reviews under secondary studies, scoping reviews, umbrella reviews. Each of them can only answer different types of research questions. So, for instance, if you are going to do a scoping reviews are usually used when the evidences within a field they are heterogeneous, and you want to map the current evidence. It's different from when for instance, you can use a scoping review to answer a question on effectiveness. A question on effectiveness is best answered with either a systematic review combined with a meta-analysis or a narrative review. Answer the question of, is an intervention effective, with a scoping review. That's not the function of a scoping review. So those are so you must know the pros and the cons, and that's why I took time to break them down. So let me just come down to the one that is our focus today, and that's what's it called? The systematic review and the narrative review. So let me talk about systematic review first. So when you say you are doing a systematic review, what that means is you are is a standardized way of answering a research question in such a way that if someone in the United States followed that same methodological framework, someone in Peru followed that same methodological framework, they are likely to get the same results. It is different opinions. That's why it is quite different from narrative reviews. In a narrative review, your opinions are you can still impress your opinion on inside the paper, but in a systematic review, your opinions are not permitted. They are not permitted. And what's the first step in doing a systematic review is to get a topic or a research question. And the popular way of getting a research question is to use the PICO format. PICO is for PICO. That's the commonly used framework to get. That's the commonly framework used to get research questions or research topics for systematic review and meta-analysis. The P stands for population, the I stands for intervention, the C stands for comparison, and the O stands for outcome. So the P is what is the population you have to look at. So I can be looking at effectiveness of, let's say, effectiveness of intravenous thrombolysis in pediatric stroke patients. Pediatric stroke patient there stands for my population. It has excluded a group of patients already, and that's the adult population. So you must always have a population target in your research question, in your especially for a systematic review. You must have a population in mind. Who am I targeting? Then what is the intervention that I'm looking at? For example, in the example that I gave, the intervention is intravenous thrombolysis, and in fact, I still have to be narrower. Which of the intravenous thrombolysis is it? Streptokinase? Is it ateleptase? You know, you just have to come down. You have to come down to you have to come down to it. So it's very important. It's very important that you come down to that. Then C is comparison. C is not always constant. You know, sometimes you can have a comparison, and sometimes it may not have a comparison. You may just be comparing it to a standard way of care. So that's it. But sometimes you can have comparison. For instance, you can be comparing, let's say, you can compare the effectiveness of streptokinase versus ateleptase for intravenous thrombolysis of adult stroke patients in Nigeria. So we already have a comparison, and we may not have a comparison. Then O is your outcome. What are you looking at in this? So in our most of the time, you are likely looking at the clinical outcome or the patient. You know, for example, in the topic now, our outcome is we are looking at the effectiveness, and sometimes you combine it with the safety of the two. So that's how you form your topics most of the time. Some other time you can add a time limit. So you can say probably within the last 10 years, within the last 5 years, it's also permissible to add that to it. So moving from that, once you have a topic, another crucial thing is, because you cannot duplicate idea, whatever you are going to do has to be novel, especially because you have to register your protocol. Once you have an idea, in fact, if you're finding your research question, research idea, go online and see if someone else has not done the same thing you are trying to do, because you are not the only one thinking about the idea you have. 10,000s of scientists every day are thinking the same way you are thinking also. So, and you don't want to refine your idea to do everything, and at the end of the day, you see that someone has done exactly the same thing that you are trying to do. So the next step is do a general search online on Google. You don't even need to go to scientific databases already. Go to Google and do your basic search. If you see that, oh, okay, nobody has done this, then you can check Prospero. Prospero is where you are going to register your protocol, and I'm still going to talk about that very soon. Prospero is Prospero. Prospero is a database that you register your systematic review protocols, your meta-analysis protocol. And the reason for checking that database is you have to cross-check that nobody has registered a protocol and they're already working on the idea. So they may not have published the idea, but they've already registered the protocol. And once they register the protocol, it's more or less like they have a copyright to the topic. So that's what I means in layman term, that once we register this protocol, is more or less like we have a copyright to that topic. So there's no need bothering yourself again for you to start repeating the same thing they are already working on. So those things are very crucial. Once your idea pass those two levels, okay, so let me just put this in. But there are times that people might have worked on the same ideas, but they may not have worked on it within the same political location or within the same time frame. So let's say someone has worked on a particular topic and the last one that you saw was 10 years ago. Obviously, you can go ahead and there's what we call an updated systematic review. That's, thinking that within the last 10 years, obviously, there are new studies that have been published on that topic. So you want to update you want to do an updated systematic review over what those people did 10 years ago. So that's still permissible. Or let's say an idea was carried out and the limitation was maybe to American countries alone or to European countries. You can localize it to Africa. In fact, if something has been done in Africa, you can localize it to Nigeria. So it's permissible, but it's not permissible that you do the same idea within the same geopolitical region, within the same study population in terms of maybe the demography and also the time frame. It won't pass. You are going to have issues at review levels, even when registering your protocol. So once your idea passed that, the next thing is to gather your team together. You can't do a systematic review alone. You can't do a systematic review. You can't do a systematic review if you are alone. That's what I wanted to say. So you need to gather your team, people of like-minded, that are ready and passionate to work too. After that, the next thing is you want to put together your protocol, your study protocol, and you want to register it on Prospero. Prospero there are other databases or registers where you can register your study protocols, but Prospero is the commonest. And when you go to Prospero, you are going to see about 30 questions that you are going to answer. You are going to answer each of them, and you are going to submit. You are going to submit your what's it called? You are going to submit your protocol, and they are going to approve it. When they approve it, it becomes your copyright. They are going to give you a unique number. But while waiting for your protocol to be approved, you can now move on to start your systematic review proper. The next thing is you want to do a proper literature search. So number one is you want to see what are the databases you want to search. So the commonly searched databases are PubMed, Medline, Scopus, Web of Science, Cochrane Library, Embase. For psychiatry focused topics, you can search PsycInfo. For African-based studies, you can search the African Journal Online Library. So these are commonly searched databases, and they usually say you should search at least 5. And definitely PubMed has to be there, Scopus has to be there. Those are the big boys in terms of the databases. So you can't say you do a systematic review and you didn't search PubMed, you didn't search Scopus. It's not possible. It's not going to pass. So these are databases that you want to search with your topics, with the topics that you have in mind, with the keywords. So, and Well, before there were not so much automated tools that can help, but these days now there are automated tools that can help with your literature review. Things like confidence. Now, confidence can help you. That's CONVIDENCE. Yeah, can help. Rayan 2 is a good topic. That's RAYYAN. So Rayan 2 can help put together all your literature search together. So you can check all of those online, and you can see YouTube videos on how to use them. They are simple or automated tools, and they've made life quite easier. So when you have your studies from these different databases, the next thing is you have to do what we call screening. Screening is, no matter how good your keywords are, no matter how good your keyword, they can be perfect in giving you the exact numbers of studies that you need. So there has to be a human overview. There has to be a woman overview. There has to be a woman overview of it, in such a way that you are going to check the titles of each of the papers and the abstract of each of the papers. They usually recommend that it is done by 2 people, so that you screen away the papers that do not align with your eligibility criteria. So your eligibility criteria are 2 things. That's your inclusion criteria and your exclusion criteria. Now, don't miss it. Your exclusion criteria is not the opposite of your inclusion criteria. So let's say, for instance, this meeting now, our inclusion criteria is medical students who have spent at least 2 years in any Nigerian school. Inclusion criteria cannot be medical students who have not spent 2 years in any in a Nigerian university. No, that's not an exclusion criteria. For example, our exclusion criteria for this meeting can be that we are looking at people who have had at least one experience doing a systematic review, or who are our exclusion, we are excluding people who have not had any experience in a systematic review. Now, that's a reasonable exclusion criteria. So when you are framing your inclusion criteria and your exclusion criteria, they are not just one and opposite of each other. You have to be careful. You have to be careful with that. For instance, let's say we want to do, like the example I give, effectiveness and safety of ateleptase in the management of acute stroke in Nigerian patients. So our inclusion criteria, definitely, we are including adult patients, that's patients with age more than 18 years, who are going to take ateleptase as their thrombolysis. A good exclusion criteria can be that patients presenting outside of the thrombolysis window. So thrombolysis window is usually 4.5 hours. That's 4 hours 30 minutes. So that's a good exclusion criteria. So that's what you are going to be measuring. That's what you are going to be measuring, like your paper against. As you are looking at each of the paper, you are looking at, do they meet our inclusion criteria? Which of the ones are we excluding? So you do all of that for each of those papers. Now, where you need all the tools that I've mentioned earlier, the confidence and the Rian, is because there are papers that you are going to see in PubMed that they are going to appear in Scopus too. All of these tools, they help you remove duplicates. So you don't have to be screening the same type of paper or the same paper almost like 5 times from different databases. They help you remove duplicates at the end of the day. So that's very, very important. So at the end of the day, you are going to have a minimum number of papers. Let's say at the end of the day, you've now saw that, okay, it's only 10 papers that meet our inclusion and exclusion criteria. The next thing you want to do is what we call a data extraction. So a data extraction, if you read journals regularly, you'll have seen some tables where they will put author and year. They will put author and year. You see them put author and year, number of participants, for study design. For example, since we are looking at a systematic review on ateleptase now, so what's the the dosage of the ateleptase that they gave the patient, what's the condition of the patient. So that's some of the things that you want to extract from each of those studies. Now, if you are going to do a meta-analysis, you need to link up with your data analyst. You need to link up with your data analyst. So your data analyst is going to give you what we call the variables that he needs to perform a standard analysis. So you are going to extract that for him. But if you are not going to do a meta-analysis, that means you stop at that your own extraction level. After that, another crucial thing you want to do is what we call a risk of bias assessment. A risk of bias assessment. So risk of biases, we want to look at the studies that we are even going to use for this systematic review. How quality are they? And there are different tools for different types of study design. For example, if the studies that you are using are cross-sectional studies, you are going to use what we call the Newcastle-Ottawa scale. That's the scale. That's the tool. We are going to use Newcastle-Ottawa scale. If they are case reports or case series, you are going to use the JBI critical assessment tool for case reports. If they are randomized control studies, you are going to use what we call a ROB2. That's it. The name of the tool that you are going to use, a ROB2. So you can just go online. There are different quality and tool for different types of study designs. So you have to, and there are things that you check. It's like a checklist. You are not stressing yourself. You are just picking the paper and say, oh, paper by Chidima et al. Okay, under the checklist, did they state the number of participants? Oh, okay, we look at it. Yes, they stated it, tick it. So they are just like normal checklist. It's nothing so difficult. So you do that. Then another crucial thing is you develop your Prisma chart. Your Prisma chart. So if you read papers, you usually see figures. There's usually a figure called the Prisma flow chart, which what the Prisma flow chart does is all the steps that have been mentioned since morning, searching database, different databases, screening them. You are not going to put them into that chart. And you can download a free version of the chart. You don't have to design it on your own. There's a free version. You can just type download Prisma flowchart. You just have to be putting your numbers in there. You just have to be putting the number. So let's say when we search PubMed, we saw 35 papers. Okay, 35 papers, PubMed. Scopus, 12 papers. Then after duplicate, how many duplicate did we remove? So it's just a pictorial representation of everything I've been saying since morning in terms of your literature search to your literature screening. That's what the flow chart is all about. Then after, you want to write your results. When you are writing the result for a systematic review, the first thing is you want to describe what's the overall study characteristics. So when we mean study characteristics, we are saying like it's just a summary of the whole paper. The crucial thing that you need to put there is, number one, how many papers finally met it or made it into your review. Let's say 10 papers. What's the total number of participants across those 10 studies? So you add the participant number for each of the studies together to get the total number. Also, for example, if we are doing something in Nigeria, we can look at how many papers are from Southwest, from Southeast, from the North, so that we'll be able to have the pictorial representation of where are the places that people are not working yet already, or is this we are even going to get? Is it general? Can we generalize it? Then after that, you want to do what is called you want to present your stuff under common themes. For example, now we say we are looking at effectiveness and safety. So under effectiveness for stroke patient, now you can be looking at what's the functional independence after 30 days, for example, that is reported across the studies. What's the functional independence after 90 days? I'm just giving an example of like sub teams. So what you are doing is across the study, you want to look at what are the common effectiveness that you found, and that's what you are bringing together as like a common team together. That's what you are bringing together. For example, for safety too, you want to look at a common issue is intracranial hemorrhage. How many studies reported intracranial hemorrhage? Also you want to look at, are there patients that later died for maybe throwing an emboli or things like that. So you have to present your result in a thematic way, in under common sub teams. That's how you present your result. If you are doing a meta-analysis, obviously your data analyst will have done the analysis part, and you are going to present that too. But if you are not presenting, what's it called, if you are not doing a meta-analysis with it, if it's only a systematic review, then you stop at that level. Then the next thing is you want to write your discussion. So in your discussion, you are not repeating the results in the discussion. What you are doing in discussion is you are summarizing your finding, you are comparing it with what other people have done before, and you are also stating what are the current study gaps and the future directions. So what are the things that you notice that are not currently being done, and what are your recommendations for the future? Then the next is to write your limitations and strength of your review. For example, a common limitation is you are not likely, because we are English speaking country, you are likely going to always exclude non-English language papers. So that's a limitation of your review already, that this reviewed English language studies. However, we know that there could have been probably other evidences too. I'm not saying that when you are doing something in Nigeria, obviously if you are doing something in Nigeria, you can use that exclusion criteria, because you expect that every paper coming from Nigeria will. But let's say you are doing something at just a global scale now, you can exclude non-English language papers because you don't have resources to maybe someone to interpret the papers in other languages. So that's the limitation of your review, because there could be evidences from Chinese paper, there could be evidences from French papers and things like that. Then the sixth thing is you want to write your conclusion. Then your last thing is you want to write a proper abstract. So abstract is just a summary of everything you've done, and you usually write it last. So that's what doing a systematic review looks like from A to Z, really. I think I should just take some questions. I know you guys will have questions. Let me take some questions and let me answer some questions. Thank you.
Sir, thank you so much, sir, for this powerful session. I believe we learned a lot. Thank you so much. Your effort is seen, and we really appreciate you. Please, if you have questions about this session, you want to know more about this, you want to ask him personally about things that you feel you are not clear with, please gently raise your hand. I'll unmute you and then you can speak. Thank you.
Please, camera, you can go ahead to ask your question.
All right, thank you, Dr. Nicholas. So my question is that when you were mentioning data analysis, you talked about data analysis. Like, is it like a part of the systematic review or it's optional? And Then also, I didn't really, when you were explaining the PICO, I didn't really understand the outcome part. And you were mentioning a lot of databases as well. So please, can you do a recap of that? Thank you very much.
Okay. So, well, usually you always combine systematic review and meta-analysis together, but it's not compulsory. Meta-analysis is just the data, a proper data analysis part of a systematic review. You can do a systematic review alone, or you can do what we call a systematic review and meta-analysis. So there is no crime. It depends on whichever you want to, whichever you want to, whichever you want to. Then PICO is outcome. Outcome is what exactly are you interested in this study? Like, what are you looking at? For example, in the topic I gave, the outcome that we are looking at is effectiveness and safety. Well, under effectiveness and safety, obviously, we can generate sub-themes under the effectiveness and safety. For example, for safety now, we can be looking at intracranial hemorrhage. We can be looking at pulmonary embolism for each of these. So we're just trying to look at the broad outcome from the topic is effectiveness and safety. But when you break them down more, you can bring yeah. So and that's what the outcome means generally.
Okay, thank you so much, sir.
Yeah, you're welcome.
Okay, please, if you have questions, please, this is the right opportunity for you to air your questions and get clear understanding. Please, if you have questions, either you type it on the group or you raise your hand, then you can speak. Please, we have 5 more minutes for that, please. Thank you. Okay, sir, there's one question for you on the group. Question is, updating of systematic review on a particular topic, can it only be done? Sir, please, did you get my question?
Okay. Is it the question in the chat box?
Yes, sir. Yes, sir.
Oh, okay.
Updating of systematic review on a particular topic, can it only be done after 10 years? Asked by Popo Olapis.
Oh, okay. Oh, no. It's not like the 10 years is not statutory. I just give that as an example. As long as you see that there is a substantial of newer evidences over what was obtainable when that systematic review was done, you can go ahead and do your updated systematic review. For example, if a study was done, let's say 2020, and they used only 5 papers for a systematic review, this is 2025. If I saw that, if I can see that, oh, the newer papers have been published on this same topic, they are now like maybe 5 or 10. So that means altogether, and I have 15 papers now, or and I have 10 papers now. That's a significant newer number of evidences. But let's say I now have just 1 or 2 papers. It doesn't make it sense doing an updated systematic review. So it's not much about the year. It's just when I use 10 years, you just believe that within 10 years, obviously, people have done a lot of newer work around that area.
Okay. Please, anyone with any other question, please. We're rounding up now. Please, anyone with any other question? Okay, in the absence of that, we want to say a very big thank you to you, Dr. Nicholas. We appreciate your efforts. We have received from you, and we hope that we put this into action, and also try to make sure that we get a right standing in research. Thank you so much, sir, for your time. We really appreciate you, and we hope to talk to you soon. Thank you so much. Thank you so much, guys, for joining the call. I hope you got something that really, really impacted you, even if it's in a small way or small manner. Thank you so much for joining. Thank you. See you. We'll meet next time. You can leave the call now. Thank you so much. Bye bye.